It's my pleasure to introduce Simon Eickhoff, who is going to be talking with us today. Simon is he's a full professor. He's currently chair for the Institute of Systems Neuroscience at Heinrich Heine University in Dusseldorf, and he directs the Institute of Neuroscience and Medicine. And I'm going to butcher this, but Forschungszentrum Jugend, I think. So,
very close.
Not too bad, yeah. And he has, got a very interesting resume in that he's kind of working at this interface between brain anatomy, data science, and brain medicine. Very interested in moving brain imaging forward to something that can be used practically, and also approaching studies with rigor and trying to look at things like individual variability, robustness, etc. So he's going to be talking to us today about areas and networks and moving towards from those to individual predictions. And so without further ado, Simon, I'll let you go ahead and give your talk.
Sorry, I apologize for a second. I'm going to mute everyone, and I would ask people to stay mute during the talk. Thank you.
Oh, thank you. Thank you very much for the invitation and the opportunity to present our work. Thank you for the very kind introduction. So, assuming that this is a somewhat heterogeneous audience, I kind of apologize to both extreme ends of the distribution, those that are very familiar with neuroimaging and machine learning on the one hand, and those that are completely unfamiliar, because I will also cater to the respective sites a little bit. But let's get started on the. So, everybody who has ever looked at any neuroimaging data knows one fact for sure. There's a lot of inter-individual variability. No matter what you look at, brain structure, function, connectivity, subjects vary widely from each other. Some of that may be noise, but obviously much of it will be driven by the fact that we are all different. There's differences like age and gender, genetics, but also aspects that we are just starting to tap into, like experience, health, lifestyle, and so on. And on the other hand, the question is then, how does the variability in neuroanatomy, in functional neuroanatomy, how does that relate and ultimately shape inter-individual variability in behavioral phenotypes, be it cognitive performance, socio-affective functions, psychopathology, and so on. And these are not new questions. There's a vast amount of literature that associates brain structure, brain function, connectivity with both age, gender, for example, and various traits. Now, the key foundation of really everything I'm talking about afterwards is that we really need to make sure what we're talking about. Are we talking about associations in a group, or are we talking about individual inference? And I will give you in the next slide a little bit of an overview for why that is such a critical difference. I guess most of you have published figures like this. You measured something in the brain in a group of patients, in a group of controls, you compare these two, and you find a nice and strong difference, highly significant. And I guess most of you would be tempted to write about the cool new biomarker you just found. Well, the tricky aspect is that the very same data, just displayed in a more honest fashion, probably does not look so convincing anymore as a biomarker. This is exactly the same data, just not shown as a summary plot, but now for the individual subjects. And I think this highlights from the very outset that mean differences or correlations across a large group are actually rather poor biomarkers, because individual features rarely allow for individual inference. That is, even if you have a highly significant group difference, there's usually no chance that this particular feature tells you something about one individual subject. Now, on the other hand, a lot of you have probably also encountered situations like I have here in my little toy example. So these are two groups, basketball players and sumo ringers. And if you look at their height and their weight in the metric system, then you see that the two groups are, no, they're not really significantly different. The p-value of 0.12, and you also see that the two values are highly correlated with each other. And I guess that's a situation that all of us, in particular those that are into clinical applications, know fully well. You have a lot of data, and yeah, you have quite a few trends, but nothing is really nicely significant, so you could write a paper about, and everything is highly intercorrelated. Now, the critical point is, if we change our perspective and look for patterns in the data, joint patterns, then all of a sudden, there's a big change. Because although none of the two values was individually significantly different between the two groups, together they allow for a very nice differentiation that extends to the individual subjects. So to really sum up this slide that provides the motivation for what we are doing and what I think many of you are doing, we have to remember that individual features are rarely useful to tell something about a single case, that we need to search for multivariate predictive patterns, and also importantly, significance on the univariate level has only a loose relationship to the importance for finding or constructing such predictive patterns. Just to juxtapose classical statistics versus machine learning, which is ultimately what it is. Once more, in the standard within sample statistics, you have one group of patients, one group of controls. That's all you have. And you're basically saying, in those patients here and those controls, is the big difference that I see surprisingly big? And if so, that's then significant. But really, clinical questions focus not on the group you've investigated, but rather they focus on new cases. So we're talking about out-of-sample prediction. And the fundamental pipeline there is that you basically take what you have in terms of data, in terms of subjects, train a model that distinguishes patients and controls, and then you test that model in new, previously unseen subjects to see whether it indeed extends beyond the patients, beyond the controls that you actually measured, that you used to train your model. So much of our work, much of, I guess, your work, much of the work in the field is focusing on machine learning for individual prediction, in the sense that we're asking a question: if I have a brain scan, can I tell something about the individual phenotype of a new, previously unseen subject? And if you want to tell something about one person, you need many. So in order to train these kind of models, you need to rely on large to extreme cohorts that are orders of magnitude above what you have for studies of within sample association. And the reason is that somewhere you need to learn that relationship between many different features in the brain and that particular outcome you want to investigate. At the moment, most cohorts that are being used, HCP, NKI, 1,000 brains, are usually in the range of a few hundred, few thousand subjects. But we're already seeing, again, a shift of pace, because now studies like the UK Biobank, the German national cohort, and really many different cohorts worldwide, they're starting to jump quickly into the tens of thousands and above. And that poses a lot of challenges that in themselves would be worth a talk, namely all the challenges that come with data management and high throughput computing. At latest with the UK Biobank, we reached the point where an Excel spreadsheet is not data management anymore. But there's another problem, a much more fundamental one. And that is what I would call the curse of pseudo-dimensionality. Now, if we look at a single scan, then we roughly at a 1 millimeter resolution have about 400,000 gray matter voxels. And if you look at these, you don't see them. You see a brain. I hope all of you see a brain here. And you can quickly make out some features in that brain. And obviously to our human mind, having these kind of images, but not 8 as I show here, but having 10,000 of these images, you would say that's a lot of data. The problem is the computer, any algorithm, does not see what you see, but the algorithm is seeing that. If you're wondering what that actually is, this is a matrix I've plotted here. And the x-axis contains the data of 10,000 individual subjects. And the y-axis, left to right, contains all gray matter voxels. Now, this looks bad. I think everybody would agree here. Of course, you have in classical statistics problems with multiple comparisons, but in particular on the topic that we are focusing in terms of machine learning, you have an extremely poor feature to sample ratio. So even with the UK Biobank as sort of the biggest data set, you have 10 times more features than you have samples. If you would say, well, I downscale to a 2 millimeter resolution, so you're downsampling by a factor of 8, then even with the UK Biobank, we have roughly, if you're lucky, as many features as you have samples. That's not a good situation. In fact, it's a horrendous situation. So it's critical to remember that we do have 400,000 voxel, but we do not have 400,000 sort of pieces of information, but rather neighboring voxels are highly similar to each other. And that is not just a function of the data and smoothness and so on. There's a lot of biology behind it. And that is, in particular, the organization of the brain into distinct areas. There's a lot of history on brain cartography. What is the concept of a cortical area? How is it challenged? And so on. I show 3 papers here that go a lot into theoretical and conceptual detail. But for the purpose of the talk, the key point is cortical areas constrain structure, function, and connectivity. So within one cortical area, features should be relatively stable, but if you move across the border into a different area, all features change rather rapidly and strongly. Show you an example for that also on the next slide. What is critical to remember in addition is that cortical areas have at best a very loose relation to brain macroanatomy. So whoever thought, oh, well, I'm safe because I'm using the AAL atlas anyways, no, you're not safe and you shouldn't use it. Because the functional and structural organization of the brain is only very loosely constrained by macroanatomical features such as gyri and sulci. Let me give you a quick example of what I mean by structure, function, and connectivity changing together. So this is a study that's already a series of studies that's already a few years old. A colleague of mine, Sebastian Bluder, mapped 2 areas in the frontal pole of the brain. So there was a cytoarchitectonic mapping study investigating the distribution and organization of cell bodies. And he found 2 distinct patterns, histological patterns. One area, this blue one, FP1 here on the lateral surface, and the other FP2 in red on the more medial part. And as you can see here already, the border does not coincide with the difference between lateral and medial, but it's rather moved about a centimeter towards the medial side. Now, we could then show that although immediately neighboring, and although they're actually part of the same macroanatomical definition, frontal pole, these two areas are involved in two very different functions. The blue lateral area is purely cognitive. It's activated by processes such as working memory, perception, and so on. On the other hand, the red, the medial area, is fully on the social affective side of things. And hence it may not surprise that they're also talking to very different partners in the brain. The blue to the IPS, lateral frontal cortex, and so on, and the red to midline structures in the amygdala. Again, if you use the AAL, they're actually part of the same definition. Importantly, we could then also show that they are not only different in terms of microstructure, in terms of interaction patterns and function, but they're also differentially affected by disease, in this case, major depression. More precisely, only the red, only the medial area was affected in major depression. Last but not least, by reference to the Allen Brain Atlas, we then also showed that there's also a difference in gene expression patterns, in particular in depression-related genes, between the lateral and the medial frontal pole. Well, I hope that this little excursion here, this little detour, has convinced everybody that it may not be a bad idea to incorporate some knowledge on brain organization into this whole process. That's exactly what I'm going to be focusing on. And most of the work is actually done by using the Schäfer atlas, resting state-based atlas, as data compression, at least sort of in the first half of the talk. So how do we go about? I'm mainly going to be talking about in the next couple of slides on predictions based on brain anatomy. There's going to be a second part that is more focused on resting state. But here now for the next couple of slides, I will be fully talking about voxel-based morphometry and anatomical features. So the idea is that you have a large group of subjects, and then you obviously need to organize and process the data. For those of you that have not heard about it, not seen it, or not checked it out, I can strongly recommend to look at DataLad. This is a piece of software developed by Michael Hanke and now also quite extensively documented with a nice handbook. And this is really, I would say, the optimal way to organize and manage your research data. After processing, we have the step that I motivated quite extensively now, which is the biological compression. So you don't take all your 400,000 or 200,000, depending on resolution, voxels, but you actually just compute the mean volume per a priori defined brain area. Then you end up with this matrix of several hundred, several thousand subjects by just a few hundred areas. So now all of a sudden, you go from many more features to samples to I have a lot more samples than features, which is something I think everybody that is in machine learning highly desires. There's another advantage that I would like to point out here, and that is that the parcellation is completely a priori. So there's no information of that particular group, of that particular subject that you have in that group, of your training group or of your test group in the compression. But you can basically take new data and completely independent of that data, reduce it to this much more handleable few hundred area format. Next steps are then to train your model on that subject by area matrix of, in this case, gray matter volumes, and then test it in new previously unseen subjects. I will, for the next 2 or 3 minutes, focus on probably the prototypical application there, and that's predicting age from T1 MRI. As you can see here, we can do that actually very well. So on the x-axis, you have the chronological age of the subjects. On the y-axis, you have the estimated or the brain age. And what you can see in this cohort, which is I think about 6,000–7,000 subjects, is that we can quite nicely predict how old somebody is from an MRI, obviously out of sample, with an average deviation of about 4 to 5 years. Now, importantly, what we've seen over the last couple of years is that you can reduce this mean absolute deviation from the original papers in the early 10s to now, but there seems to be somewhat of a lower bound, in particular when you're talking about generalization to new cohorts. And the question arises whether this is something where we're just not good enough in terms of the algorithms. Well, most likely not. But what seems to be the case is that this is a true variability. And it makes a lot of sense, right? Because we are trying to predict chronological age, but what we're actually predicting is biological age. So not how old is the subject, but how old does the brain look? And it stands to reason that there is a lot going into that, in particular, a lot that relates to individual health and lifestyle. And so it's not surprising that if you look at subjects with mild cognitive impairment or even Alzheimer's disease, you see an increase in brain age. In particular, subjects with MCI have about 5 years advancement, and Alzheimer's subjects almost 10 years. This has been well established. And so what we thought is, okay, we are focusing on a new topic, but we do not want to conflate new algorithm, new subjects, new questions altogether, but rather we combine the question that we're interested in, which is here, sleep breathing disorders, which is mainly obstructive sleep apnea, a few other ones. We combine that with a replication of the well-known dementia effects. So here's the raw data. If we go into a bit more detail, we have the healthy controls. Their mean brain age deviation is about 0, which it should be. If we look at patients with MCI, we're finding what everybody else finds. Yes, we have a roughly 4-year advancement. But interestingly, neither in the patients with mild cognitive impairment nor in the healthy controls, there was a significant difference between patients with sleep-related breathing disorders in black and those that did not suffer from it in gray. What happens in patients with actual dementia? The same. We again replicate the findings on the brain age advancement, but we don't see any difference between the two groups that were formed by sleep disordered breathing. So what I would take from that, and what I would also recommend, I think, in terms of a trust building exercise for a very quickly growing field, is to combine new questions with replications of old questions that I think will solidify what we think we know, but it would also lend a much higher credibility to somewhat surprising or null findings. And that finding was indeed somewhat surprising because from the cognitive literature, it's very well known that sleep disordered breathing is a risk factor for dementia, that patients with this disorder have a worse cognitive performance than those that don't have it, and so on. But this is apparently not reflected in BrainAGE. Another application example that I'm going to quickly go into, because I'm going to focus here a little bit more on some technical aspects, is Parkinson's disease. So we had 2 larger cohorts of Parkinson's patients in healthy controls. That's the PPMI data set and some from our local collaborators. We again used the very same approach in terms of processing and representation tested for group differences, and as you would unfortunately expect from the literature, it's somewhat diffuse and not really giving you a very strong focus of atrophy as you have in Alzheimer. And then we also wanted to predict brain age. And to do that, we had quite a large reference cohort. Well, in fact, it was several cohorts, as you see here, several large public data sets. Now, what you also see in this histogram is that there's quite some bias. There are some data sets that contribute a lot more older subjects. Other data sets contribute a lot more younger subjects. So if we would just train our model on all of that data, we would be proud because we train it on so many subjects. But we also, unfortunately, buy in quite a bit of bias because age and sight and all the effects that sight brings with it were highly correlated. So the idea that we followed here, that we actually also used in the obstructive sleep apnea paper, is to perform a stratified subsampling. So we built an ensemble classifier where each weak learner in the ensemble was perfectly balanced in terms of sight, age, and gender. So each individual model only saw a small proportion of the training data, but that proportion of the training data was completely unbiased. And then basically we combine the predictions from all of these small learners, weak learners, into a joint model. And if we test that, we can actually see that this allows us to predict individual age in the healthy control subjects very well. And in fact, it fits extremely well with what we would expect, like a 4.5-year mean average error and very little bias. Now, what happens to the Parkinson's patients? What we see here is quite a couple of things that are, I think, each in itself worth mentioning. First of all, the effects are not so big compared to mild cognitive impairment, compared to Alzheimer's, but also compared to disorders like schizophrenia. Parkinson's patients have a relatively moderate advancement in brain age, which is just about 3 years. And that is actually something that lends itself to a lot of discussion. I can't go into all of the aspects in detail, but basically it raises the question whether the idea that neurodegenerative disorders are merely an advancement of the physiological aging process is up for discussion. There was a small advancement, but importantly, it was replicated across both cohorts. Last but not least, we also see a quite marked inter-individual difference with quite a few subjects also having brains that look actually younger than they really are. I'll come to that in a minute. So we have consistent differences that are smaller than MCI and Alzheimer. And what's important then, they also relate to clinical phenotypes. In particular, we see that there is a significant correlation with the disease duration. Now, if you wonder about the colors, the red one is the local cohort. The orange one is the PPMI, which is a progression initiative. So basically all the subjects were very early stage. Moreover, we also found that there was a relationship to both the UPDRS-3 score, so the motor impairment, and the MoCA score, the cognitive impairment. To sum this subpart up, it seems that using the BrainAGE approach, we can find rather robust effects. Think about the replication of the Alzheimer and MCI findings in our sleep disorder cohort. Think about now here the replication of the Parkinson's findings in two different samples. So that's good. BrainAGE seems to be fairly robust and replicable. But there's also quite a few open questions that relate to inter-individual variability, a lot of variance around the mean, and also relates to the fact that effects of neurodegenerative disorders may be surprisingly small relative to the actual impairment of the patients. Last but not least, we also tested whether we can find an association with cognitive performance in normal aging. And here we looked at the Benton score, classical neuropsychological test for brain damage. And this really reinforced the picture. Yes, there are fairly robust differences, quite robust, but they are small and they're accompanied by a lot of individual variability. And hence, what I think our main challenge now is for the next couple of years is to leverage the very large to extreme scale data sets to really go into depth in terms of all the different factors, health, age, lifestyle, and so on, that will probably jointly affect brain aging and decline. So now I talked quite a bit about brain structure, but we obviously know that brain structure is just one aspect. The brain is a dynamic network of functional interactions. And so it obviously makes a lot of sense to also consider these features to see what can we gain in terms of individual prediction by looking at brain networks. And again, I'm building on the same fundamental idea as before, but in this case, the critical thing here really is to relate what we know about brain function and cognition to our individual measures. Brain function, how do you measure it? Well, you measure it with fMRI. That one is trivial. But I would argue that whenever we measure the brain by resting state MRI, we get a nice window into individual function, into individual fluctuations. But obviously, we cannot really relate what we're finding to functional systems, mental processes, and ultimately cognition. Now, that's not a problem because we have task fMRI, and task fMRI shows us what brain areas are involved in, for example, working memory, what brain areas are involved in reading, and so on and so on. So it stands to reason, it's probably quite intuitive that we could use functional MRI data to basically define our brain networks and then in new subjects look at something that's easier to acquire, such as resting state, in these networks. And that's a great idea, but it's also a somewhat tricky idea. Why is that? Because ultimately, I'm not sure how many people in the audience do task-based functional imaging. Task-based imaging suffers from a lot of problems. Most effects are non-replicable. You have a lot of analytical flexibility. The NAPS papers showed that very nicely. There's also many different ways of how you can define a certain function for investigation. And I won't even mention those things that nobody should do, like selective reporting, p-hacking, hacking, and so on. So what can we do? How can we define our this is the set of brain areas that are involved in, let's say, working memory? Well, we have a very noisy, but also, and that's important, a very big and standardized literature. So the idea is to consolidate this big, noisy literature into some robust network models that then can serve as a biological prior, as a biological data compression for individual data analysis. I'll show you an example here of neuroimaging meta-analysis. And that's a study we published quite a few years ago now. So here you see the activation foci reported in about 200 papers on working memory. And what you see is that basically they're scattered all over the brain, but more likely in some parts than others. We can then use techniques such as activation likelihood estimation to quantify the convergence after accommodating spatial uncertainty, and then perform statistical inference against a null of random spatial association. And that brings us from dozens, hundreds of papers onto a robust network. So these are the areas that are across subjects, across scanners, across paradigms, across analysis pipelines. These are the areas that are robustly associated with working memory tasks. That's the key thing. We are not sort of we are not staying in the resting state, but we really want to know what are the areas that get truly active, more active than rest, when you do a particular type of task. And then we can conceptualize the peaks of this network sort of as nodes of the relevant network, and then extract individual resting state data in these networks. So we go from the broadest of all levels, hundreds of papers, thousands of subjects, towards a robust network definition. And then we use that basically as an assay to then extract individual single subject resting state information in exactly these networks. Is this information useful for prediction? Glad you asked. Here's an example where we tried to predict personality traits. So what you see on the left is the emotion processing network, and what you see on the right is the prediction of individual neuroticism in new subjects, obviously, from the network connectivity in or among these areas. And as you can see, we can do that reasonably well. So this is a correlation, I think, of 0.3 something. So that's good. Per se, the idea works. But somebody may argue, well, maybe you can just test anywhere in the brain. Maybe you always get this kind of prediction. Well, let's test that. Let's look at two different networks. So here you have the cognitive network, cognitive action control, on meta-analytically defined. Here on the right, you have a socio-affective network. And then we extract resting state in these two networks. Now here comes a small, but I think extremely critical piece of information. It's the same resting state scans. The subjects were only in the scanner once. There was one resting state time series that has been acquired, but we're basically extracting different parts of the same data based on a priori knowledge where the different networks are. And if we then train regression models to predict, for example, a cognitive score, here the Corsi block tapping, we see that it does indeed matter, and it matters a lot where you are in the brain. The cognitive network allowed us to predict the individual block typing score very well, whereas there was no predictive capacity at all in the socio-affective network. Last but not least, I want to now wrap a lot of the things that I've set up and show you one application example in somewhat more detail that really shows how these different bits and pieces would work together and give you hopefully a better picture on brain organization and pathology. What you see here is psychopathology information on patients with schizophrenia. More precisely, that's the PANSS scores from about 2,000 individual patients recruited at 10 different centers worldwide. And big thanks here to you, Vince, because part of that was the Cobra sample that I think you were involved in when you were in New Mexico. Now, to be a bit more precise, we actually had one big sample that came from a intervention study in Northern Netherlands, the famous data set with about 1,500 subjects. Good, nice, clean data set. And we had 500 subjects. That was the worst mess you can imagine. I wrote here 10 centers. One yielded 1,500. And basically the other 500 came from 9 different places: US, Germany, rest of Europe, Asia, different clinical systems, different diagnostic systems, definitely different raters. It was a pure mess. Why was that good for us? Well, it's good because if you think about Punt's factorization, then those of you who have a little bit of insight into the field know that there have been tons of papers that immediately, after 1 or 2 years, were refuted. You can just not get a good factorization. So we turned the whole thing on its head and said, well, we want to find a factorization. We want to find a dimensional structure of psychopathology based on the fact that it's robust and generalizable. So we had our big uniform data set, and we had the still quite big 500 subject chaos data set. And then we tried to factorize the data and find the solution that best transfers between both parts of the data. And there we ended up with a 4-factor solution. And since we used, in this case, the OPNMF, so it's a sparse, non-negative factorization, they're actually very well interpretable. So the first factor represents negative symptoms: lack of spontaneity, social withdrawal, motor retardation, and so on. The second is psychotic symptoms, delusions, hallucinations. The third is actually quite interesting because it's been discussed quite a while, but never really made it into the public mind. Somewhat of a more effective part: anxiety, guilt, feelings, tensions, and so on. And last but not least, a bigger factor that represents cognitive and disorganized symptoms. So what we have at that point is a 4-dimensional structure of psychopathology that is very well generalizable across many different data sets. And on top of that, it's also highly interpretable. If you are getting interested, and yes, I'm teasering here, you can actually use this because we have a website where you can just plug in your Punt scores and get the dimension scores back. Next, we tried to cluster patients into distinct groups on these 4-dimensional representations. Why is that? Because this factorization is obviously not only reducing dimensionality, but it kind of also acts as a denoising. And we were really hoping to find several distinct subgroups in this very clear and interpretable space. Well, turns out we did not. We found 2 subgroups: a positive psychotic type and a negative type. We were surprised. The reviewers were even more surprised. They were so surprised that they made us compile a 62-page rebuttal letter because they really wanted us to test everything whether we cannot find more groups. But they also wanted a lot of other things. Long story short, no. There are only these 2 subgroups. Now, are these subgroups also different in neurobiology? We investigated this by using the Schäfer atlas for data compression, but then not inputting the whole connectome, but actually rows of the connectome. Now, what is a row of the connectome? A row of the connectome basically is the connectivity pattern of one brain area with all the other areas in the brain. And then we fitted and evaluated a classification model, positive, well, this negative subtype, for each of the brain areas. And what you see here now is the classification accuracy of the rows, more precisely, the classification accuracy of the connectivity profile between that brain region and all other ones. And what's very interesting to note is that this region-based classification actually performed quite a bit better than whole brain classification. Now, is that possible? Because obviously, the whole connectome includes the row in question. So why are we better just using one row than using this one row and everything else? The answer is very simple. Unfortunately, we have not 100,000 patients. So ultimately, it again comes down to feature to sample ratio. So reducing connectome-based analysis to one row at a time gives us not only a better predictive performance, since we can now achieve a better feature-to-sample ratio, but also a interpretability, because we now know which brain areas are most predictive. And if you look at the top hits, you can actually test on what they are doing. And if you do this, for example, here for the right TPJ, you see that this area is involved in social cognition, theory of mind, and face perception. What do I mean by involved in? Well, this is being activated by task fMRI studies. And I think this link between task fMRI and resting state analysis is something that is much too small in the current field. We need to strengthen that link, because both have a very unique and very say intriguing potential to add to our understanding of the brain. Sticking with task fMRI, remember I said we can have the very same resting state data set, but then use different networks as data reductions. Well, we do this here as well. So we have a group of schizophrenia patients, and we look at a range of different meta-analytically defined brain networks. So these are the areas that are involved in affective processing. These are the areas that are involved in action control. This is semantic memory. This is motor control. So we know what each network is doing, and we extract just the part of the resting state connectome in that one network. What we then try to do is to predict on the individual level the loadings of these factors. So basically, we want to predict individual dimensional psychopathology. And these are the results. So what you see here is that there are quite a few associations that, or quite a few network by symptom dimension combinations that are predictive. Now, some of you may think, yeah, well, if you have a big trawler, you will find some fish. You're right. This is, at the start, a big fishing expedition. It yields candidates. So what did we do? This is based on a, first of all, tenfold cross-validation. In the next step, we then pruned our candidates to those that actually also allowed, that also yielded a good leave one side out cross-validation. So only if you could significantly predict in each of the left out sides, we kept them. And then we really went into the cold water and said, okay, well, I said practically to Justin Baker and Avram Holtz, give us a new data set. It's the BSNP data. And this is, again, from a different site. It's in New England. And it's actually also, that makes it even a bit more challenging, next to COBRA, the only other data set that is North American. So what happened? We look at the top network here, the social effective default, and its predictability of the cognitive factor. We can see that among the 6 combinations, see them on the bottom here. Can you see my mouse, by the way?
Yes, yes.
Okay, good. So there were 6 network by symptom dimension combinations that were significant both in the cross-validation, the tenfold, replicated tenfold, as well as significant for each individual site in the leave one side out. And if we tested these 6 combinations on the BSNP data, boom. So there were 5. Boom. 3 of them failed completely. Again, we required that it worked in both other sides. And so ultimately, there were just 2 combinations of networks and symptom dimensions that survived all the successive steps of pruning. And I think that's exactly where we go when we talk about biomarker discovery. We need to screen a lot of potential assays. Like all of our networks are specific assays into the resting state. We screen a lot of them. We throw away a lot of them. We have another validation step. We again throw a lot of them away. And then we really need to take the hard road, take completely new data, give it a shot, and again, throw away a lot of them. But I think only this approach will ultimately lead to something that is really applicable. And if you ask, well, how good is the prediction? This is what we end up with, and that's after adjusting for age, gender, disease duration, and so on. So we actually end up with a correlation between true and predictive of 0.3. If we do not adjust, it's obviously better, but that's kind of trivial because disease severity really drives everything. Finally, I talked a lot about prediction, and I love prediction, but we also want to get some insight. And we did that by using the use-based toolbox, which again is publicly available. So to relate node predictive performance to receptor PET, and we could show that you not only have that predictive capacity of one network, which is actually generalizing across all the different challenges, but it also relates to the distribution of dopaminergic and, interestingly, serotoninergic tracers. And the serotoninergic is really interesting because, as some of you may know, cognitive symptoms are not really well ameliorated by dopaminergic medication. So we hope that the kind of stream that I showed here, from massive screening to successive refinement to hard evaluation, in combination with this sort of more mechanistic insight, that will, in the long term, then lead to new clinically applicable classifiers and also provide new leads in terms of mechanistic treatments. So I hope over the last, it's about 50 minutes now, I have convinced you that mapping the human brain, knowing where to look in terms of areas and networks, modeling inter-individual variability in a predictive manner, and working towards personalized brain medicine are 3 very closely intertwined challenges that we need to address together. With that, I would like to close by thanking you for your attention, particularly, however, also thanking our funders and everybody I had the pleasure to work with over the last couple of years. And thank you for your attention. I'm now more than happy to take questions.
Thanks, Simon. So we can take some questions now. There were a few questions that came up in the chat. One of them was, how do you address the fact that most of the literature reports positive findings? So basically, dealing with the bias, of the sort of file drawer problem.
You mean in terms of meta-analysis?
It didn't specify, but I think, yeah, you can maybe decide.
Yes. So let's say the positivity bias is not so much what I'm worrying about. What I'm mainly worried about is conditional positivity bias. I'll give you an extreme case. You are using an effective Stroop. You cannot start to write the manuscript and publish a paper before you show me amygdala activation. And the poor student is sitting there and sweating and running analysis by analysis until they find the amygdala that the supervisor really wants. And only then they write the manuscript. This is something that is a real problem. But there's a good side to it. The good side is that neuroimaging is quite expensive and quite time consuming. So ultimately, I think the vast majority of findings at the end sees the daylight somewhere. That's much worse in other fields like psychophysics or cognitive psychology, reaction time data, where it's very cheap to put a undergraduate on collecting some data, analyzing it, doesn't work, you just leave it in the drawer. That seems to happen rather rarely for MRI data because it's so expensive. Now we know that there are biases. We can't get completely around them, but the overarching idea is that hopefully there's enough that's being published somewhere so that it, to a large degree, cancels out, and those things that are robustly observed, they then come up in the meta-analysis. Our biggest weakness, as I said, is biased positivity bias.
Hi, Simon. This is Yuping from Tulan. So you make a age prediction mainly based on T1 MRI image using gray matter volume. So how about using a functional connectivity network? Or can you combine both of them? And what would be the added value for using both functional connectivity and gray matter volume? And furthermore, looks like, the methylation data can also be used for predicting ages.
Yeah.
Can you also incorporate genomic factors in the prediction?
Yes. So I'll start with the last one. We haven't done it yet, but of course you can. The question is really, and that then circles also to the first question, what do you want? Basically, age prediction is quite well feasible by several different features. So I would, I think, more be interested midterm in understanding what drives the differentiation. And if you combine, let's say, gray matter volume and resting state data, what you see is that there can be some improvements, but it's not massive. The one thing I like about using the structural imaging data, the resting state, the VBM, is that we do have all the variability on the personal history side, genes, gender, lifestyle, health, and so on. And if we then use resting state, we would also have all the variability on that side, right? Because what are people thinking about individual fluctuations and so on? So I think focusing on something that is less state-specific, namely structure, is a good avenue to understand all the trait influences and the interrelationship of trait influences. Another aspect that is very relevant here is the fact that T1 is something you can also get, also post-hoc, in a lot of clinical cohorts. So this makes the translational aspect a bit easier.
Okay, thank you.
A related question on the chat, what's the ground truth in age prediction? Our ground truth is, you should probably put that in inverted commas. Our ground truth is chronological age, but we know that chronological age is actually not the ground truth for BrainAGE. We need to make the assumption that on average, biological and chronological age is the same, but we know that in our training data set and obviously also in our test data sets, this is something that at best holds on average, not on the individual level.
So for your age prediction, so what are the population that you use? Are you only focusing on the aging populations? So have you tested on those adolescents or children to study whether they have a delayed brain development or accelerated brain development?
So we have just worked on aging because, well, that's the focus in terms of content. We're already doing too many things probably. We will hopefully work in development from actually very early age on in the near future if we get funded for that. But other people have worked in development, adolescence in particular. Like there's a lot of work in particular from UPenn on BrainAGE and adolescent development, and the picture is very similar, basically. So you can predict it reasonably well. If you have a more narrow range of prediction, if you just look at, let's say, a 10-year span in adolescence, the accuracy is higher. You're getting like 1.5 to 2 years rather than 4.5 years that you can do, that you get with several decades. But really, everything I said in terms of aging also holds for development, from what I know, from what other people have done. I haven't done it myself.
Okay, thank you.
I have a question. So to what degree do you consider, when you're looking at sort of parcellations, right? Overlapping parcellations, sort of, you know, in which the degree of overlap maybe varies, right, from individual to individual. How important do you think that is? Or do you factor that in at all?
There are, I think, two answers there that are somewhat contradictory. The one is we are at the moment mainly working, or almost exclusively working with group average atlases like the Schäfer Atlas, and we're seeing that there are also across modalities, across many different applications. I think there's half a dozen published papers and many more in the pipeline. They work well and robust. We also know that individual mapping, that overlapping, accommodating, can enhance performance. In fact, it does enhance performance. Why am I hence not fully embracing on the individual parcellation idea as people like Hasheng Liu are doing? Well, the reason is that I'm somewhat of a skeptic and somewhat of a person that is very much interested in pragmatic application. If you look at clinical grade data, then the theoretical advances of individual parcellation quickly break down. Because if you have poor and short data, then your individual maps get more and more noisy. So at the moment, at least, much of our approach is focused on using group average atlases, knowing that they're not perfect, but give us quite some degree of robustness. And the next step that we are now looking into, both in terms of atlases and also in terms of meta-analysis, is whether we can allow some flexibility in terms of accommodating individual expressions thereof.
Great. Yeah, that's something that I think is quite valuable, is sort of leveraging the group, you know, the denoising aspect of the group information. And I think you referred to this earlier as sort of, you know, using it as kind of a prior or as a way to sort of regularize the solution. So you allow for both individual variability and you can leverage these sort of large N studies.
And I think that's particularly important when people start to realize that they may have moved themselves into a corner with the HCP dataset. So I'm seeing a lot of fascinating modeling on HCP, always in the discussion about patients and healthcare and so on. HCP is both the best and the worst data set that you can work on. It's the best because everything works. But it's the worst you can develop your methods on because the quality is so high that you have no guarantee whatsoever that your beautiful method also works in other data sets.
Yeah, double-edged sword. Okay, I want to be sensitive to the time. We're about 5 minutes past the hour. So I guess if we can maybe take one more burning question.
I can take
yeah.
some more questions. It really depends on you, and then I don't want to hold everybody up. Just to quickly jump in here, I'm more than happy to take any questions also later, offline, by email, social media, whatever.
Okay. We'll probably connect you in with the Brain Space Initiative discussion.
I am already.
Perfect. Okay. So we'll announce that as well. So thank you. Any other questions?
I have 2 questions, but I will ask on the Slack. No, right. Or via email.
Are you scared everybody away?
Not taking advantage of his position with the mute button. So. Let me just ask one more, maybe as our last question. And this is actually Armin's question, but I think it'll be relevant for other people as well. When you were estimating those factors for the PAN scores, can you estimate those indices using other variables, not PAN's variables? Have you tried that? Is that something that would be interesting to look at?
We haven't really. We've focused almost exclusively on the PANs, but it would be extremely interesting. So one thing that, I'm just gonna move back a bit here. So one thing that we have in this paper that will come out very, very shortly is a try to basically relate this prediction performance also to other cognitive measures in the BSNP group, and it works reasonably well. So what I think is that there is a potential for also translation between different psychopathology scales on this side of sort of machine learning, on the side of brain networks. We haven't tried it yet, but the main reason is that we didn't. Actually, the BSNP data is the first data we really have that has something else than PANS, at least as well. But what we are lacking is a big data set with ideally resting state and structure that has detailed, let's say, sub-SUNS measures. We just don't have the data. If you have that, you should definitely hook up.
Yep, definitely. Thank you.
Simon, could you please spell out your email address?
I can actually type in the chat. It's probably easier. I can't type.
And just as a reminder that the talk is recorded and it will be shared at the links that Armin has sent via the chat, and also that will come out later via email and via the Slack channels. So
I'm just posting my Gmail address because all professional addresses forward there anyways. So.
Okay, yeah, keep life simple. It's already complicated enough, right?
Okay. But particularly at the moment.
Yeah. Well, thank you, Simon, for joining us. This has been great.
Thank you for hosting me.
A lot of impressive work, and I think you're really moving the field forward. So
thanks.
Thank you very much. And thanks everyone else for joining. And we will stay in touch going forward.
Yes, we should definitely. Well, thanks to everybody, and enjoy the day.
Thanks. Take care. Bye-bye.
Bye.